I'm going to go ahead and get started. So hello, everyone. My name is Adam Farsheed, and today I'm going to be telling you about my cool research. I'm also going to give you a little intro about like who I am, because I've learned that if you know a little bit about the speaker, you tend to care more and pay more attention. And so I always make that a point of mine. Okay. So first off, who am I? I went to Laguna Beach High School between 2009 and 2013. I played football and soccer, and I went on the current list of teachers, and I see that I have all of these teachers listed here that are still at the school, which was cool to see. I would say that my favorite subjects were probably math and physics. I was just kind of into math, physics / stuff, and that's why I chose my major probably. And then I have some embarrassing photos here. So this was after I got into Northwestern. I wore like this full kit of stuff. Yeah, it was weird. And then over here, that's like, if you look me up, that's one of the photos that pops up. So when I was a teacher, all my kids were like, is this you? And I'd be like, yeah, that's me. And then down here, I have a picture here. There's Miss C2 from last week, actually.
Adam.
Yeah, I know. I know you didn't want to show it, but it's too bad. But basically, we all were in ACR together. This, I think, was at like formal or something. And we all did like the right-hand rule from physics. So yeah, it's kind of lame, but it's okay. After that, I went to Northwestern between 2013 and 2017. I majored in mechanical engineering and minored in philosophy. Go Cats. And I think I majored in mechanical engineering because my friend from high school, he was majoring in mechanical engineering, and he said, you should do that if you like math and physics. So that's how I came to major in mechanical engineering, which is probably not the best way to do it. After that, I was in a program called Teach for America, which is basically where you work for 2 years as a teacher in a school that basically does not have enough teachers and resources. And so I was placed in the state of Washington. I did that from 2017 to 2019. And while I was there, I taught 7th grade math, 10th grade geometry, and 7th and 8th grade PE randomly because they needed another teacher. And then in 2019, I came to Rice. I just finished my first year at Rice. So I just finished my my second semester. I'm waiting for my grades to get back. Hopefully I do good. And here I'm getting a PhD in bioengineering. One kind of interesting thing about me is I kind of went from mechanical to bioengineering, which basically means that I took no biology in college. So last biology I took was in high school, and I also only took an intro-level chemistry class in college. And yeah, now I'm here learning all that stuff as I go. So I think this is to say, like, you can kind of change what you want to do later on. But it was extremely helpful that my research that I did in undergrad was relevant towards bioengineering, and that's kind of why I switched in. So I have this map here to kind of show my journey. So I started in Laguna, then I went over to Evanston, Illinois, then I was in Tulsa, Oklahoma for the summer after I graduated, getting trained to be a teacher. So that summer I did my teacher training. From there, I lived in Seattle, but I taught in a city called Kent, Washington. And now I'm in Texas. So next I just have some pictures about college. So some organizations that I was a part of, some clubs in college I was a part of, that I very much encourage you guys to get involved in college clubs as well, because I didn't do that until the end of my first year, which was a big mistake because I didn't even know what it was. But one club that I was part of was called Engineers for Sustainable World. And we were basically a bunch of mini clubs with different projects, all working towards sustainability as the end goal. So I became the co-president of that club, and I work specifically on a project called Hydroponics, which is where you try to grow plants without the use of soil and with less water. So it was a cool thing. I had never heard of it before. It was a little engineering, so that was fun, and I got to meet some cool people. And here I have basil and Swiss chard that we grew back in Evanston. Next up, I was also part of the Special Olympics Club on campus. So what that entailed is I volunteered every week for all 4 years. with the local Special Olympics chapter. And I basically got to know the athletes well, played sports with them, did like some cooking randomly with them. But overall, it was just a really great experience and really impactful for me. And here's us. We got to go on the football field and say hi to all the fans at the middle of a game or something like that. So that was pretty cool. I'm back here, both hands up. And then finally, here is what the weather looks like sometimes in Evanston. So unlike Laguna, there's a lot of snow. My first year there, it was called the polar vortex because there was such a storm going on. And I think like I had been to the mountains before and been in cold environments, but it's different when that happens for like 3 months straight and you're trying to go to class and you have to walk in this. And yeah, it's just a lot of new stuff. So next, I'm going to talk quickly about my Teach for America experience. My school was called Excel Public Charter School. It's a charter network that's in California, also in Tennessee, I want to say, and then they were in Washington, but unfortunately they pulled out of Washington. I think this year they pulled out. So I taught there for 2 years, and it was really sad because at the end of my second year, we had to actually shut down our school. And I had to tell our kids, basically all my 7th graders, that they couldn't come back to the school next year, which was honestly extremely hard to do, and something that I really never envisioned myself having to do. And just to give a feel, this was what my school was. It was inside of a church. We didn't have a dedicated building. And so our gym was actually right in the middle of the school. If you can imagine like where the quad is at Laguna, our quad was a gym and everything that entailed. And this classroom right here actually was my classroom. So it was not uncommon for a basketball to hit up against the window, which, as you can probably imagine, if you're teaching a group of 7th graders, it's not very conducive to some great learning. But we had a great time. This was my first class that I taught. So these were my 7th graders. The school was 7, 8, 9, and 10th grade, which was odd, but it is what it is. So this was my classroom with the Northwestern University Wildcats. This is my classroom I just have a couple photos here. So these are some of my students. I had a cool 3D printer in the back there, if you can see that. Here's me and my friend Mustafa. We took a selfie. He moved like abruptly, so I think this is like I took a selfie before he was leaving. And then finally, I have a picture of me and the soccer team. We didn't have a soccer team at our school, but I formed a soccer team with some other charter schools. And we played 2 games. We were 2-0, so I'm undefeated. There's a great sign here made by my girlfriend. And our jerseys don't really look that official, but we played really well. And then I have a couple of cool or funny videos. This is one of my students, Farah. I think this is pretty self-explanatory. So Farah thought he was strong, and then I did some dancing with my students that I thought you guys would maybe think funny. The song says bad words, so taking it out. Yeah, but we definitely had a lot of fun at my school. The last thing I'll just talk about is on the last day of school, my first year of teaching, there was a student, Jack, who you can see in the back has really great hair, and my hair was getting really long, and he said every day, Mr. Farsheed, can I cut your hair? Mr. Farsheed, can I cut your hair? Mr. Farsheed, can I cut your hair? And I said, okay, fine, Jack, you can cut my hair. And then this is what happened. So don't let 7th graders cut your hair. That's the end of that. So before I move on, quickly pause for any questions about anything I've talked about so far. And next up, I'm going to be talking about 3D printing, my 3D printing.
Okay, students, this is your opportunity to type questions in the chat, and the teachers will be monitoring the chat window. I'll start as you think about questions to put in the chat. How did you like teaching, Adam?
I loved teaching. I thought that it would be a lot easier than it was, and I thought that because I was good at math and physics, that I would be good at teaching math and physics, but that is completely wrong. I think that because I was good at math, I it was hard for me to break down all of the individual steps. And I think that I didn't expect to, when I was going into it, I didn't think about like, what will I gain out of it? But I think that I gained a ton of interpersonal skills. And also, I think I'm pretty good at breaking stuff down now. I'm not sure if I was before, but I like to think that's from teaching. I gained that ability.
Yeah, the gifted, I'll call you a student, sometimes does have a hard time teaching because you're thinking, oh, it's obvious, isn't it? And
Right.
No, it isn't. And so then you have to think, well, why is it obvious to me and not to you? And that's the art of teaching.
Especially 7th grade concepts that
that
I would think are easy.
The development of the brain is an interesting thing.
Yeah.
Okay, but it looked like your class sizes were reasonably small. Is that true?
Yep. The class sizes were fine.
And is that typical of Washington, or was it a special school so that?
I think it was more so because our school was a public charter school. So basically, it was open to anyone, but someone had to go out of their way and enroll their student in our school. And so I think because of that, I don't know. I don't think we had a clear image because what our school ended up becoming was actually a school with people on like both sides of the spectrum in terms of like their old learning experiences. So we had students who were like excelling in their public schools and they wanted to join something special. So they joined our school. And on the other end, we had a lot of students who were basically like kicked out of their old schools and they needed to go to school somewhere. So it was a tough balancing act, but it was a great experience.
Yeah, very interesting. Yeah, because when I visit other places and I say, oh, my classes are 30-something, and they say, really? Ours are 20-something. And so California has big class sizes. All right, here's a question for you from Chantel Carter. Chantel is class of 2018, maybe. So she's an alum. How did you apply for your PhD program when you did not major in biology as an undergraduate? And what do you think helped show your ability to succeed in a bioengineering field?
Yeah, that's a great question. I think that, so I studied mechanical engineering and I applied into bioengineering. So that's pretty different. But the good thing that worked on my side was that my undergraduate research was basically a bioengineering project. And so because of that, I could basically say that like, this is what I've done and this is what I want to do and sound somewhat competent, even though like, I don't know, I mean, still to this point, I don't know any biology. Like I was in cell biology first semester. But again, like the last time I took biology was in high school, like not even senior year, like before that. So it was, I don't know, it's interesting. I get to learn a lot of new stuff, which is cool.
Yeah. Okay, so we'll get to your research real soon. One more question before we do that. What made you want to become a teacher, even though that's not what you majored in college?
That's a good question. I was actually like recruited, so I didn't know what I wanted to do after I graduated. I knew that I probably wanted to go and get a PhD, but I did want to take a year or two off, which was a recommendation that my mom gave me because she got her PhD right after she graduated undergrad, and she said, don't do this. So I didn't do that. And then someone emailed me and was like, hey, I'm a recruiter for Teach for America. We see that you're interested in giving back to the community. We're wondering if you'd be interested in this. And I had done some tutoring, some private tutoring for a company actually for like 3 years, 4 years maybe, while I was in high school. So I thought that I knew something about teaching, but I didn't really. But it was a fun time.
All right, excellent. Let's continue with the talk now, and we're going to learn about 3D printing research.
Awesome. So, so my project, I think it's summed up in 3D printing custom biodegradable stents. So when you work in a lab, or if you want to work in a lab at a university, you basically are working for one of your professors in their lab. And so what professors do is they spend most of their time trying to get money from various sources to fund their lab for certain projects. And so basically that money that they get for these projects funds you as you work in their lab. So they're kind of like your mini boss, even though you're still a student, which is kind of interesting. And so this was my mini boss, Professor Chang Sun. He was a great professor to have as my PI. I was really lucky to have him because he really, from the get-go, let me get super in-depth, and then also like kind of included me on all of the research that was going on. So I was really grateful for that. So I did this between 2015 and 2017. So this was my junior year summer, and my senior year. And so one of my 4 classes was lab class, where I basically there was no specific times, but I would just come in whenever I had to do something, and I would work in conjunction with a graduate student who was like more high up and was like, hey, we should do this, or you should do this. And he was kind of like my mini boss. And my junior summer, I was able to get a grant to fund me while I stayed on campus. So all of these things are kind of things at most universities that you should definitely look forward to or look out for when you go to college, if this is something that's interesting to you. And so I worked on 3D printing stents. So I'm going to break this down into 2 regions. First, I'm going to talk about stents, and then I'm going to talk about 3D printing. So first off, I found this cool graphic. Hopefully this doesn't like get messed up when you see it. But this is showing that within an artery, you can have a blockage, such as like if you have a buildup of cholesterol, or in this video, it says there's plaque. It's narrowing the artery, the amount of blood that can go through. And so that's bad because blood needs to get through, right? So the procedure to help with that blockage is you go starting from the arm all the way through the blood, all the way into wherever the blockage is. And you put this really small tube called a catheter. And you load this really small metal device called a stent. And it's used to push open the walls to keep it from collapsing further. So here they have, I mean, this was just a random video, but most of the time this is done with a metal stent. It's usually made out of a material called nitinol. That's kind of like a springy material. So I think of like a slinky, but in a circle. So if you like compressed it all together and then you put it in the right spot and then you allow it to kind of go out. And so in this video, they showed a balloon pushing it open, but a lot of times you want it to naturally spring on its own towards the wall. So you don't need a balloon. It just kind of by its properties, it springs out. It wants to go back to its normal position. So what are some problems with stents, right? Why did I care about this? So first of all, stents are very expensive to manufacture. If you can imagine, if you're going to put a metal piece in your body in a vein, and it's also very small, maybe a diameter of 3 millimeters, which I have a ruler here. It's like this. From the end of the ruler to here, it's pretty small. They're very expensive, and also you have to make sure that they're polished and really nice so they do not rupture any walls in an artery or something like that. So that's one big thing. The second thing is that it's kind of a one-size-fits-all mentality. So kind of like small, medium, and large shirts, right? There'll be a small, medium, and large stent. And the only thing that might change is also there's like a 10 centimeter long and then a 20 centimeter long or whatever it may be. So different lengths. And so that's a problem because everyone's arteries are not the same. There's so many different uses for a stent. So it doesn't really make sense to use this sort of one-size-fits-all methodology. So the third problem that I've identified is there's some long-term problems. So if you have a metal stent in your artery, maybe 10 years later, if that thing is still there and it gets dislodged or something and moves down an artery, you know, that can cause a lot of problems. And then the final thing is that in pediatric cases, there's growth in the patient, right? So if you have a child that needs a stent, and that child's going to grow and their arteries are going to grow, that's going to also cause some major problems. So then the motivation came, let's create a stent that's cheap, customizable, and biodegradable. So cheap gets problem number 1, customizable fits this number 2, and biodegradable is going to fix number 3. And so the idea is, and it's been shown, that you don't need a stent to hold open your artery for 10 years. After about, I think it's around 6 months, it's been shown that the blood vessel will naturally kind of take that new shape on. And so the idea is, what if we can just put one there for 6 months and then it'll biodegrade away? It'll just go away. So I have some pictures here to kind of define scaffolding. It's all after this idea of scaffolding, and a lot of times instead of stents, we use the term scaffold. That was kind of adopted as a newer term. And so for the Empire State Building shown here, you can see that there's these chunks of scaffolding at each part in the process that kind of aid in the final building of this building, but they aren't part of the final structure. And so that's the whole idea. Can we aid the body to open its artery up on its own, but then after that, go away and let it do its own thing? So next, I want to talk a little bit about 3D printing. And I found this graphic here. There's a bunch of different types of 3D printing. Do not care about the slide. All I'm trying to say is you can print many different ways and also with many different materials. So you can use plastic, you can use collagen, which is like a part of the body. You can use metal, you can use concrete even, right? There's so much you can do. And 3D printing is more of like a general like idea of like, what if we try to do 3D printing as opposed to like one specific way to do that 3D printing. So for example, like there are many different types of computers, right? But not all of those computers work exactly the same way. So this is fused deposition modeling. Is probably the technique you guys are familiar with if you've heard of 3D printing. And so the idea is basically that of a hot glue gun, but with plastic and at a much finer precision. So you have this bulk of plastic that's hard, and then you heat it up, just like in a hot glue gun, and then it becomes liquidy, comes out of this nozzle. You can create a certain arrangement, and then quickly it cools. And then you can put your build platform down a little bit, and then you can do that again. And you can build layer by layer and build a cool 3D object. But that's not the type of 3D printing I used. I used something called stereolithography. And this is a technique that works by completely different mechanisms. So it's nothing like the hot glue. Instead, you have this resin, which is basically just like a liquid bath, a specific liquid. And what happens is you shine a specific type of laser, a UV laser, or actually other types of lasers too, at the bath. And the bath, basically, there's a chemical reaction that I'll show on the next slide that occurs, and that liquid becomes a solid. And so what you do is you will, as shown here, like you do this first layer, then you have to go up, the laser will go do its thing, then you go down into the bath, then you go to recover it, then you go up, you laser again over and over until you can get this sort of 3D shape. And I want you guys to notice here that in this method, you're going down into the bath, okay? Because that's gonna change pretty soon. And the reaction that occurs is called photopolymerization. And so what it is you have a liquid with multiple different components, but in its most simplified form, you have some sort of thing, some sort of monomer or one chemical formula that you have, that's what it is. And then you also have this photoinitiator, that's another chemical that's in the bath. And when you shine a UV light onto this bath, the photoinitiator, the UV light carries energy. And I actually remember getting politely scolded in Mr. Sogo's class about not covering myself while I was using a UV light because it's not good for you if you look at it or if it touches your skin. And the UV light basically activates the photoinitiator. And then there's these chemicals that are like, I want to react. And then all of these individual pieces get linked together through links, through covalent chains. And so you get this solid, right? This is now, they're all linked together. So you can't really move that. Whereas this one, if you pushed like right here, this would maybe move over here, right? So it's a liquid to a solid based off of the UV light. And so there was this breakthrough called continuous liquid interface production a few years ago that was the basis for what my research specifically was on. So I'm going to show you a video from this company because they make it look really, really cool. And then I'll explain what's going on after. So this is 7 times speed. So this is going on faster than they're showing, but it's still really cool. Whoa. I'll skip forward a little bit. Basically, there's this thin bath here. Now there's a big 3D printed part here that does not fit in the bath. So it's like, what? How'd they do that, right? It looks like magic. So the way that this works, it's based off of the previous technique I talked about, where there's the resin that's being solidified. In the previous slide, I said that it was moving down into the bath, but you can see here that this isn't moving down, this is moving up. It's pulling the solid out of the liquid, which makes it look like 10 times as cool as well. And so the way that that can be done, which is a new and a novel idea, is that you allow a tiny bit of oxygen to pass through. So you have this UV light source that's projected. Sorry, I went too fast. So you have a UV light source. That's the UV light. You shine it at this DMD. What a DMD is, it's the same as a projector in your classroom. What it is, it's like thousands, if not, thousands of tiny mirrors that are either shining towards this direction or just like reflecting the light somewhere you don't care about. And so what that is you can put in a program and say, I want these specific mirrors to be shining. And what that does is it creates from just a broad UV light a specific pattern that you want. And so that pattern of this UV light is shown into the bath. And you have this oxygen permeable window at the very bottom. So what this oxygen actually does is it prevents the photopolymerization reaction from happening. So the reaction I was talking about earlier. And so why this is so important or cool is that you can basically, instead of doing it one at a time, go down, up, down, up, down, up. For a small piece that might be the size of your pinky, there'd be like 4,000, 6,000 layers. So you have to go down, up 6,000 times, which is going to take a long time. Instead of that, you can just move up at a continuous pace, and you can just cycle between each of the images that you want to print. And so you can basically, and I'll show it a little bit better on the next slide, come up with a model and then print it layer by layer. And so, again, the critical thing here is that this oxygen prevents the reaction from happening. So it starts over here. So if you pull up, you can just pull it directly out of the bath. If this wasn't here, this would stick to the bottom and this would not work at all, right? You would just have the object would be printed at the very bottom and then you're trying to pull it off of the glass, which is stuck to now. So the whole cool thing about this was the oxygen that allows you to go a lot faster. And so how we design something, right? I have this cool thing here, like what is that? Well, we take a software called CAD, which is a computer modeling software, somewhat similar to those that are used for like Pixar movies, I would like to imagine, or animated films. They use a similar software, and we can create a certain shape that we want. So this is the shape that we want. This would be very difficult to build either through old methods or through using the fused deposition modeling types of 3D printers, just because of some of the geometries on it. And what we do is we basically take like a electronic deli slicer and we slice this into 4,000 individual slices, and then we get 4,000 cross-sectional slices of this thing. So if you played starting from here all the way to here through the system, you would get the full shape by the end of it. And so we can stack all the layers on top of each other and get our final shape. And so one final graphic to kind of tie it all together is we start with this specific design that we've created on our own, and we can change that and kind of do whatever we want. We then feed that through a computer into our DMD chip is really what's doing most of the work, right? That's moving the mirrors to say, what do we want to reflect and what do we not? And then we have this platform go up slowly. And then after a few minutes, or maybe many minutes, you can see this beautiful picture that my professor took of that 3D printed part. And then we can do further analysis, like, let's see how good did we do as a job, right? We defined this to be 150 microns here, and it's 155.4 microns. So it's not perfect, but it's pretty good. And so one thing to note through all this is what's cool about our system is we're printing really, really, really small, minute things that you really can't do with other methods effectively. And one thing that I didn't mention before, but I'll mention now, is that with this system, this clip-based system, where you're moving continuously up as opposed to down, up, down, up, down, up, this part that we printed, which is about the size of your, maybe like half your pinky, but not as wide, would take 11 hours to print a really good version of that on the down-up version, on the old printer. But it took us around 11 minutes to do that on our new printer. So basically, you start the print, and then 11 hours later, you say, oh, cool, the first layer didn't even work. I just wasted 11 hours waiting. As opposed to now, we can go in the lab and say, oh, the first layer didn't work. Don't worry, I only wasted 11 minutes. So that was a really cool thing about this. And so, and I'm talking about it a little bit more, but one of my projects was like, we need to make this system work well, right? I just explained to you this brilliant idea, but it's not just like, oh, you know, you just click, 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 and it's done, right? You have to come up with the software, come up with how fast are we going to move the stage, how much intensity are we going to use for the light, and also keep in the back of your mind that we wanted to print parts, this stent, that had similar properties to the metal stents that I talked about earlier. So we wanted it to be kind of like slinky-ish, which is not easy to do with plastic. And on top of that, we also wanted it to be biodegradable. So that's what we were working towards. So here's some pictures of the printer, so what it looks like actually. So here is the DMD chip. This is what's doing the mirror projection. The UV light is like under this stuff. And then here's a, looks like a camera piece. We actually have a camera here so we could see what was going on. And then right here, this small little guy is actually the bath where we put the liquid, and this was the stage that went up. And I'll show just from another angle, so this is looking from the other side, kind of these cool optical components. So you would take the UV light, you would shine it, it would bounce basically off of the DMD chip all the way through here, underneath, bounce up underneath the bath, and then it would cure in the bath. And we also had this camera here so we could basically see what was going on. And then I have a video of what our printer looked like. So again, this is sped up. It's a lot, in my opinion, I mean, I think people still think it's cool, but I think it's a lot less cool than the clip one, just because we're printing something that's so small, it's hard to see what's going on for most of the process until the last couple seconds.
So there's already plastic forming, solid plastic forming under the surface of the liquid.
Yep, we can't see it right now until a couple seconds from now, but yeah, there's, right now, stuff is happening, you're gonna have to believe me, besides the stage just moving up. And again, this is very, very small. This is about like the size of the end of my pinky. It's probably the size of the part that we're coming up with.
Okay, so now we can see the solid being pulled out, but the actual reaction's happening maybe a centimeter below the surface?
Yeah, it's much lower. It's underneath the bath.
Neat. There we go.
So there's our part. It still had some liquid resin, so if you dab it off with like a little Kim wipe, you can see what this final part looks like. And this took, I think it said 9:30. And so again, it went from like 11 hours to around 10 minutes. Like, you're pretty happy about that when it happens. And so I spent specifically before, like, what was I actually working on? And so what we wanted to find out is we wanted to say, what parameters do we need to use for this to work? So there are two major things. And so this structure on the right was used to determine both the light intensity that we wanted to shine and also the speed at which we moved our stage. So what we did is we shined a certain intensity of light, and down here, we moved slowly And then at each of these individual things, we went faster and faster and faster in small increments. And then after that, we could take our part that we got, we could image it with really fine precision images. So again, this is 1 millimeter, which means that this structure is really, really small, right? You can't just see this with your naked eye. And we could measure, say, how big is this thickness? How big is this thickness? And at what layer was the thickness correct, right? You can imagine that at this bottom layer, when you have way too much of this light than you need, it's gonna just start curing random stuff that you don't want. And at the top, you don't have enough light to cure what you want. And so you kind of have this trade-off where it's like, I want to get this part to have the correct dimensions. And that's kind of the end goal that you're measuring. So this is one method we used. Another way we could use the same sort of structures, we could do, instead of changing the speed, we could change the intensity and have the same speed throughout. And we could say at which point is it overcured, undercured. And then second, what I was a major part of is we did a lot of mechanical testing. So as I explained before, we wanted these to be the same, if not better than the metal nitinol slinky-like stents. And so the way that we would test that is we wouldn't just poke it and say, oh yeah, it looks the same. We'd have to do some pretty in-depth testing. And so what that looks like is you can see I've put a stent in between these two metal plates. And what you do is you very, very slowly move this top metal plate down, and you measure the amount of force that this stent is pushing back up. So you can imagine that if I put a piece of wood under there, right, and you're pushing down on that wood, it is pushing back really, really hard, right? Wood is not going to like to be compressed. Whereas if in another example, I put a bowl of Jello, just the Jello under there, if I'm moving down, the Jello is not pushing that hard back up, right? It'll be deformed pretty easily. And so what we did is we would push down to about half of its diameter, and then we would go the opposite direction. And at that same speed, we would go back up. And we would still measure how hard is the stent pushing out. Because again, we want to have that stent pushing out so that when we put that in an artery, it pushes out towards the artery. It doesn't just sit there. So this is the whole kind of idea behind our project. And a lot of our validation was like, are we even close? Like, what's going on? So just a quick overview of what we were able to accomplish. We did printer optimization of the light and the stage. One thing that I've completely kind of ignored up until this point is we also had to come up with the liquid we were printing with. That's not just something you can buy at the store. It has, I think our B ink, as we called it, had 5 different chemicals in it. And we had to basically say, like, what if we have more of this and less of this? So if we have more solvent, it's going to be a little bit less, it's going to be less viscous. So it's going to be more like water. But if we have more, then it might be more viscous, then it's more like honey. So all of that sort of stuff has to come into account when you're basically pulling something out of the liquid. If it's like honey and you're trying to pull something out of honey, it might pull back. So there's all these different factors you have to take into account. And kind of the main 3 things that I kind of said we can confidently say is we were able to create a 2 centimeter tall stent printed in 11 minutes. We had a 150 micron wall thickness. So I didn't really talk about that either, and I don't want to go too much into it. But basically, we couldn't just make our stent bigger to make it stronger. We had to make it still thin enough where it could be compressed, but also strong enough. And also, we were able to get higher stiffness than nitinol. So if we were to put a nitinol stent under that compressor and that same design of our material under that compressor, we could get higher stiffness than the nitinol by our measurements, which is pretty cool. And so the question is like, now what do we do when we did this? Well, what you do in science is you try and publish it in a journal and tell everyone else what you did. And so we had 2 different publications that I was a part of. So this first one was mainly about this material, this cool material. And I didn't really work a lot on this. This was more a bioengineering lab, actually, that we paired up with. And they came up with the material, and we were working mostly on the printer side. And so they were reporting this bioresorbable, antioxidant, photopolymerized, whatever that word says, stent, right? And they were also saying that the mechanical properties were similar to nitinol and that they were self-expanding. So cool, awesome, that's a publication. And so what you do is you list out all the people. You can see there's Chang Sun. I'm back here because I was an undergrad and I really didn't do a lot for this paper. I'm honestly even happy I was included in on it. And then most of my bulk work went into this paper, which is mainly all of what I just talked about is kind of in this paper, but a little bit more specific. And so I worked like pretty heavily on this, and I was able to be the second author. And Henry, actually, I'm still friends with Henry, and he just graduated with his PhD like a couple days ago, maybe. Yeah, so it's pretty cool. So just wanted to say, like, you don't just return a report in at the end of the day. I want to say, like, what do you do when you have this stuff? Well, you try and publish it, and then you have people that know what they're talking about, like, hey, either I believe you or I don't believe you. And if your stuff is reliable, then it can be published in journals. And so this is called the peer review process. So it's where like some people read the papers of other people and they say like, do I believe this? Or is this just like, they're just saying stuff that's not true? One quick thing before I pause for questions is that our stent was also self-expanding through temperature. So the idea here, before I show you the video, is what if you could compress our stent to a small diameter and you could freeze it so it's really, really cold, and then it holds its shape. And then you put that stent all the way through the arteries into the blockage. The body's naturally warm. It warms it, which makes it self-expand out. And then you have your stent that does its job. So here I froze it into that U shape. And then if I'm just trying to drastically show the reaction happening, if I heat it up, it pretty quickly straightens out into a straight line. So this was at the later end. This was after we published everything. We're just trying to say, like, what other cool ideas that we have that we might be able to do. And so, so this is about 25 seconds in, and then after a minute, you can see that it straightens out completely, which was the original shape of the object. So with that, I will pause for questions again, and then next I'm going to be talking about my current research.
Okay, so once again, students, try to put some questions in the chat. So, question for you, Dr. Farsheed. You've made the stent. How do you test it in a living animal?
That's a good question. So basically how biomedical work usually goes is, and it's a little different than the mechanical side that I was a part of, but how you would test that stent is first you would say, like, is this safe at the most basic level? So what you would do is you would just take that stent and then you would put some cells on that stent and you would say, like, are these cells dying because they hate this, or are they like somewhat okay with it, or what's going on with cells? And so let's say you put it with cells and the cells are happy and everything's great. And then you're saying, okay, cool. Well, I'm going to go on to the next step. I'm going to actually try to use this in an animal model. So with just cells kind of in a petri dish, kind of like I think Lisa was explaining, that's called in vitro. And then the next step up kind of to the validation process is called in vivo. And so that's where you would actually, in an animal, you would use a stent and you would say, you know, what happens. And so you might even publish those findings. And then after that step, you would have to do some safety clinical trials where you would use these stents and you basically just say, are these safe? Like, not even are they fixing the problem, but just generally, are these safe in human beings? And then after that point, you would have to then do another set of studies to say, are these working better than what's currently available? So there's a long process there, but it's cool to work at the very basic part because you don't have to worry about all of that paperwork.
All right, here's a question from Clarice Curris. How did you get the opportunity to work for your professor on this project?
That's a good question. Really what I did was I took his class. I got an A in his class. I thought he was a nice teacher. I looked him up. I found his lab website. I thought the work sounded cool. And then I sent him an email saying, like, hey, this is who I am. I took your class this time. Here's my CV. I would love to work in your lab. And Chang Sun was generous enough to respond and say, yeah, more than welcome. And then he met with me and he let me be a part. So it was actually really, really easy to join his lab, which is why I'm so thankful that I was lucky for this opportunity.
So how many people total were in the lab?
There was about 6 people in that lab.
Okay. Not too big then. That's great.
Yeah.
All right, here's a question from Harley Feroz. What chemicals did you use to make your printing material?
That's a great question. So I'll flip back. So this paper. So this paper, they created this methacrylated poly 112 dodecamethylene citrate. I don't think I've ever actually said it before. I called it MPDC. And so that was the specific chemical that can be biodegradable and also is bioresorbent, and it's also antioxidant, and that's what was created by the other lab, and that's what the monomer was. So that's like the individual building blocks that I talked about originally in the photopolymerization. We also had to have a solvent, such as like ethanol or something similar to ethanol, if you've worked with that, in our thing. And all that did is it made it more viscous. So as I was saying earlier, if you had honey and you added some water and you were able to mix it all up, it would be a little bit easier to mix it than previously. And so we wanted to have a liquid that wasn't too sludgy, but it was able to be printed, basically. We also had to have something that was the photoinitiator. So that's a different chemical that when UV light shined on it, the reaction occurs to cross-link everything else. And then we also had to have some UV absorber, actually. And so what that did is that made it so you need it set a lower boundary on the number of UV, the amount of UV light you needed for the reaction to occur. Because if you had no UV absorber, then if you shine some light in a certain region and it kind of like deflected a certain way, it might cure something that you don't want to be cured. So you actually want to set a bottom threshold on like, I actually want this much light for this reaction to even occur. And so those are basically the components that we had.
Okay, I was wondering about that because it needs to be really precise, and I'm imagining if you just shone light, then you get plastic forming where you didn't want it. Okay, so there's this, you need a certain intensity for it to work, and so all that's part of the chemical mix.
Yep.
That I guess you didn't play with that, but clearly other people in the lab made modifications.
So we were given the MPDC, but we actually kind of messed around with the different concentrations, like how much solvent, how much MPDC, and that sort of thing to come up with our final concentrations.
Okay, very good. Very good. All right, I think we'll let you go forward and talk about this next area of research.
Okay, so should I plan to stop in 4 minutes? Should I go super fast or what?
Take your time, do it well.
Okay.
And if students need to leave, they'll drop out on their own. But we've got a good crowd, and we hope they'll stay to the end.
Awesome. Won't be too much longer. So I'm just going to briefly touch on my current research. So this is, I forgot to change the title, but this is working on multi-domain peptide hydrogels. I put it down here, of course. So that is what my new work is focused on. I'm working under Dr. Jeffrey Hartkrink, who's pictured here. Again, I'm at Rice University. I'm wearing a Rice University shirt, go Owls. And I started in 2019, maybe finish in 2024. So this is actually going to be longer than I spent in undergraduate, or for your guys' case, I'm going to be more time here than you guys will have been in high school. So it's a little crazy that I just started on this process, but I'm up for the ride. And so quickly, I want to explain to you guys what I'm working on, but I'm going to start at a very basic concept, which is what hydrophilic or hydrophobic molecules. So hydrophilic means water-loving molecules, and an example is water. If you have, I pictured here, multiple water molecules, if you have them all next to each other, they have these hydrogen bonds that form between the oxygens and hydrogens of different water molecules, and there is this attractive force between them. So that's why water will form a liquid and stick together. Now, in contrast, there's hydrophobic molecules, which are water-fearing. An example is olive oil, which consists a lot of triglycerides. So this is the structure of that. We're just going to ignore this left chunk and just look at this. All of these little thingy-majiggies are carbons connected with hydrogens. So it's just carbons and hydrogens. And the reason that they just show these is because chemists are lazy. One thing I should mention is I'm now in a chemistry lab, which is a full 180 of what I thought I was going to be doing, but that's how it goes. And so you have these molecules and these hate being next to water. That's not true, 100% true, but we'll just lie and say that that's true. And so what happens if you take olive oil, you mix it with water, you'll see the water really wants to be next to other water molecules, so it kind of pushes all the hydrophobic olive oil to the top. And olive oil is like, okay, well, I don't really care if I'm next to other olive oil, so I guess I'll be together. So it looks like these are attracted together and these are attracted together. In reality, the water just really loves itself, and then this stuff doesn't really matter. And so this is called the hydrophobic effect. And this is kind of like where you might have seen it, right? I have this leaf with some water on top of it. And you might ask yourself, like, why does water do this? Why does it form these circles? And it's this idea of surface tension. So if we zoom in on a water molecule, you have a water molecule, for example, in the middle. Well, this guy is happy because he loves water and he's surrounded by water. But if we go to one on the outside, he loves water. Well, he has water on the inside, but not on the outside. So he wants to be attracted towards the middle. So if you can imagine, all of these guys on the outside are like, I want to be towards the middle. And what that does is it forms a tight water, this little circular structure. I have a video here of me explaining it, but we're not going to go over that. So next up, I'm going to quickly talk to you guys about amino acids. You might have heard this term before. But basically, these are the building blocks of proteins. So all proteins are made up of combinations of tens or hundreds or maybe even thousands of these amino acids in certain sequences that make some protein have a certain function, which is pretty cool and mind-boggling if you think about it. I don't really believe it, but I guess it's true. So we have different types of amino acids. Some are positively charged, some are negatively charged. Some have hydrophobic side chains, kind of like here, like I showed earlier, these like little V thingies. These are all hydrophobic. On the other hand, these are hydrophilic. So there's a lot of different types, and together, these different forces, whether it be positive, negative, hydrophilic, hydrophobic, are used to create different proteins. Well, what if, in our lab, we try to say, what if we could create a multi-domain peptide hydrogel? So what if we could just use those amino acids as the building block, and we can build cool, smart structures? So in our case, I'm just going to quickly go over. This is one molecule that we've been using. It's called K2SL6K2. So the K stands for lysine. Those are on either end. The S is the serines, and these all end up on this end. The L's end up on this end because it's an SL repeat. So you go LS, LS, LS over and over. And so you get this aggregation of hydrophilic on this side, hydrophobic on this side, and positive charge on either side. So you might say, okay, cool, you showed me that. Now what? Well, what's really cool is if we put that in a solution of salt water, and I'll explain why salt water is necessary, you get this hydrophobic packing. And what happens is you have one of them that orients itself in the direction I just showed. And if you can add another K2SL6, it would try and flip over, and it would want the hydrophobic sides to be close to each other and the hydrophilic to be separate. So it's kind of similar to the oil and water. The hydrophobic part wants to be next to the hydrophobic part. So these would stack together, and you could get this really long chain formed. And so if you add this to some sort of salt solution, you get the formation of a hydrogel, which is basically like a jello, but for purposes of medical research primarily, along with other stuff, I guess. And the reason it has to be a salt solution is because I mentioned earlier, you have all these positive charges on the side. And as you know, if you try to take two positive charges and put them close to each other, like this guy and this guy, they don't like that. So the only way that they'll go ahead and form this is if you have negative charges that are in the solution, and they kind of like will go and like mask, kind of cover all of these positive charges with negative charges. And then the molecule's like, okay, well, since that's covered, I'll go ahead and form a fiber. And what's really cool is the idea is you can take something that's in a non-salt solution and it's in a liquid form. And then once you put it in a salt solution, it could become a solid. So if you injected this into like the environment within the body, it would go from a liquid in the needle to a solid as soon as it hits the wound site. And if you look at this structure on a very, very fine, under very, very fine precision, so this is 300 nanometers, which is, I wrote it down, it's 0.0003 millimeters, which is basically to say it's extremely tiny. If you zoom in on this gel that's shown here, it stays as a gel, you see this nanofibrous structure, which is very similar to the extracellular matrix that makes up the body. So a lot of our body actually also has a nanofibrous structure. And then a couple more slides, and then I'll be done. What we can do is we can inject this into an animal. So this was done into the back of a rat. And we can say, we can take a thin slice of it. So this is what it looked at the beginning. And then 6 weeks later, it looked like this at the end. So you can see that, well, it's degrading away, which is kind of something I was talking about earlier. And if you image and you stain specifically for cells and other things, you can see that all of these little dots are cells. And what's cool is by day 3, week 1, week 2, you can even see more and more. These cells are rushing into the hydrogel, which is a really good thing. You do not want the cells to be like, I don't like that, I'm not going to go there, okay? Because that's some sort of response that the body doesn't like. What's good about this is that the cells are like, oh, I recognize that, maybe because it's amino acid based. I'll break it down and I will go ahead and I'll start regenerating that region. Regeneration might consist of putting nerves in that region. Regenerating might consist of putting blood vessels in that region. There are many different ways that that might be the case. And so you can see that the hydrogel area goes down over time, and this region can be regenerated. So finally, last thing about my project, I'm working specifically on volumetric muscle loss. Basically, this is a problem where usually if a muscle has tears, it'll naturally regenerate because it doesn't, because it kind of knows what to do when it still has its extracellular matrix. But if you have a volumetric muscle loss, which is a large wound, such as maybe a huge traumatic injury, maybe it's an explosion in the Iraq War, something like that, your muscle can't regenerate that region. And so the idea is, if you take this large chunk out, what if we could put our hydrogel to basically help the muscle regenerate itself to go from injured to back to normal? So that's kind of what my project will be doing. Maybe I'll be doing 3D printing. That's something that I'm looking into now. But again, I'm at a very early stage, so I can't say as much about what I'm going to be doing in the next few years. So finally, my advice for you is get involved in research as an undergraduate and find a lab that looks interesting and just go ahead and email the professor. Any final questions as I show you a picture of my new puppy, Marshall?
Okay, students, once again, try to put some questions in the chat. So it looks like, Dr. Farsheed, that at the end there, the scaffolding with your stent kind of applies to what you're looking at here. Is that sort of what drew you to this lab?
Yeah, so I was very, very interested in biomaterials from my undergraduate research. And even though I was working on the 3D printer, I got very interested in like, hey, I don't understand the biology or the chemistry or any of that. And I kind of want to know more about that. So that's kind of why I switched over. And so, yeah.
So in your current lab, is it mostly chemists or biologists or?
Mostly chemists. I'm one of, I think, 3 bioengineers. So my other lab also works on much more chemistry-focused stuff, such as like synthesis of collagen. But this is specifically what I'm working on, and they know a lot more chemistry than I do. But I know a lot more mechanical engineering than they do. So.
Right. Yeah. Yeah. The interdisciplinary nature often makes for great progress. Okay. Here's a question from Alex Mork. He picked up on the amino acids part here, and he's wondering, what is the connection with creatine monohydrate and amino acids?
So I don't know what creatine monohydrate's made out of, but I do know what it does, and it's an energy source. So I learned that this semester, which I thought was pretty cool. So just in a 1-sentence summary, maybe, in the same way that ATP has energy that your body uses, creatine is another form of energy, and it's more of like for short bursts of energy. You breathe so that your muscles work. But I'm pretty sure creatine is it goes through a process that does not need oxygen. So your body has a bunch of ways of using energy or taking energy out of different molecules. The most common one is you breathe in oxygen, you breathe out CO2. So that goes one cycle. Creatine is another cycle that I don't think includes oxygen.
And this will probably be the last question. So again, you're starting in a test tube with the intent eventually of getting to in vivo in a living organism. As far as the lab itself goes, are there some that are on the in vivo experimentation stage?
Yeah, so actually, one thing that's like very real now that wasn't real undergrad is like working with animal work actually. So I'm intending to, and I'm currently now like figuring out what do I want to do, or what sort of procedure am I going to have to do in vivo. So within the next couple months, hopefully, if everything goes to plan with the university opening up and stuff, I'll actually be working in vivo with animals, looking at some sort of similar experiments to the one that I showed with these slides here. So it was already, this K2SL6K2 has already been used in animals, and it's already been shown actually to help nerve regeneration and also to help angiogenesis, which is the creation of blood vessels. So the K2SL6K2, it's really cool because I'm kind of inheriting this biomaterial that's already been used really well. And I'm trying to think of like, what are new ways we can use this, or can we make a new sequence potentially? One thing that I'm currently really interested in is like, instead of having this nanofibrous hydrogel that's kind of going in all directions, I want to try to make it so it's all aligned. So if you can have an aligned scaffold, you can put that in a muscle and the cells will say, hey, that's aligned. I know that I got to go that direction. And so to strategically align it so that you can help regenerate certain injuries.
Okay, and that's maybe where your 3D printing can do that, huh?
Right.
Make it all line up.
I think 3D printing is in my future again with this work. I'm just not sure how long until I get there.
Yeah, okay, that's wonderful. All right. Thank you all for participating in today's webinar. I will be closing this meeting soon. Special thanks to Dr. Farsheed for the excellent presentation. So we'll have another webinar next week given by an ACR alumnus. So hope to see you there. We had a total of 160 something at our peak. So thank you all for coming and hope to see you again sometime soon.
Thank you.
Thanks.
Thank you. Great job, Adam. That was awesome.
You.